scans to make a feature. If you have really wide peaks, you might want to have more scans. And this is something that you just have to kind of get familiar with your data. And in our case, we'll put 6. So we know for our data, 6 is good. Again, out of the box, 4 will probably be fine. But it really is up to you, the expert of your own instrument, of your own data, to just try and play around with the settings. So there's a lot of other parameters that would be really tiring to try to manually fill out. So one of the settings that we can try to extrapolate other information from is this full width half maximum. So again, just so you can see, the full width half maximum is, let's say you take a peak, is this value right here, where it is the width of the peak at half the maximum of its signal. So with this information, what is this full width half maximum? You can get information about other parameters used in Mzmine without actually having to put it there manually. And in this case, again, default settings works quite well, and we will keep it default. The last thing to consider, and this is probably one of the harder things to I think, wrap your head around, is RT tolerance intrasample and RT tolerance sample to sample. So, if you guys are unfamiliar with Orbit Trap is a high resolution mass spectrometer. So we're able to get not only the peak of a specific metabolite, but we also get the peak for the isotope, the C13. Intrasample retention time tolerance is how much does the retention time shift between the two isotopic signals within the same sample. So if we're looking at the C12 and the C13 peaks, how much do the retention time shift of the two isotopes within the same sample? You can see right here, the retention time shouldn't shift at all if it's the isotopic peak. So it should be actually a quite conservative, strict value, and the default settings of 0.04 is quite fine. The retention time tolerance between sample to sample is now, let's say you have one metabolite in 3 different samples, and it's the same metabolite in 3 different samples. Does the retention time shift between each injection? It can shift a little bit. Again, this is really up to you to know how well your instrument is conditioned, how well is the state of your current column life. And again, default settings worked quite well for us, 0.1. So that is it with the Orbit Trap, the UHPLC. Next, we have the Orbit Trap settings. So before, we were setting all the parameters needed to help define what is a peak in chromatography. Next, we need to now tell the instrument, or the Mzmine, what are intensities that we're interested in, and masses, of course. So we're just going to keep this no filter. You can set it to positive if you want positive. You can set it to negative if you want negative. You can keep it no filter. Mzmine will detect it on its own. We analyzed our data in positive mode. I forgot to mention that before. I just keep it no filter. Mzmine does a great job detecting on its own. Next thing. This is something that you can keep default. You have factor of the lowest signal, or absolute intensity. What is this, and what does it mean? So, not all signals from the mass spec are going to be meaningful. Some of it is just going to be noise. And we need to tell the Mzmine what is that level of noise. So we can either use factor of the lowest signal, so basically what is the lowest signal found in a given scan, so let's say 1E3, And if we made it a factor of 5, you would say 5 times 1e3. So anything with a signal of 5e3 and below would be considered noise. And this is nice because it then considers each scan and it defines the noise level for each given scan. Absolute intensity, on the other hand, will just set a noise level throughout the entire run, regardless if the noise is slightly different in one scan or another. Today, we will use absolute intensity, and we know with our instrument, we can set 1E5 for our MS1, and for our MS2, we can do 2E3. Some of you might be asking, how do I know, how do I determine the noise? For the sake of time, I'll not cover it at the moment. If we have time at the end, I will be happy to show you. Otherwise, there's a lot of documentation. The documentation page on this is very, very good on explaining it. And again, if we have more time at the end, I'll be happy to go over it. Excellent. So again, just to really stress what we're doing, we're trying to tell the software what needs to be considered when looking at each individual point to group bunches of points together to build a peak, i.e. again, a feature and or a metabolite. So we've just told the software what signals are noise, junk we don't care about. We now want to tell the software what signal is something that is meaningful. So this setting right here is what is the intensity required to really say, hey, this signal means that it's actually coming from something that we're interested in, some actual meaningful feature. And again, it's going to sound really annoying and redundant. You have to know your instrument. Each instrument behaves differently, especially not within its own from one orbit trap to another, but really when you start using an orbit trap to a QTOF to something else, you will have different intensities just based on how well your system ionizes. So we know we can use 1E6. And so what I really would recommend to do is, if you are unsure, is just for those who were at the first session that I showed online, and for if you weren't there, I'm assuming that you're familiar with how to inspect raw data. Just open up a chromatogram, open up a couple chromatograms of your analysis, and try to find a specific metabolite or feature that you're interested in, create the XIC, and maybe take a look at that XIC and the XIC of the isotopes, and that will give you a really good idea what settings you should use for these heights, and also this is true for any kind of retention time shift or mass tolerance, which is next. So, just like with chromatography, you could have some kind of retention time differences between injections. You can also have fluctuations in the MZ values. So again, ions are going to the mass spec. It's scanning, 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 scanning. It's not perfect. It's going to have small fluctuations. And how large those fluctuations, again, really depend on your instrument. How well is it calibrated? Something going on, somebody injects some really dirty stuff now and you have a lot of ion suppression that's causing it to be a little bit more wonky than usual. So again, you need to check it on your own, and each instrument, depending on if you're using a high resolution, medium resolution, or low resolution, will affect these numbers. And again, when you select the presets, it will give you default settings that should match a general instrument that you're using. So again, for our purposes, we know that the scan-to-scan tolerance, the intrasample tolerance, and the sample tolerance defaults are working fine. And again, I think these are some of the harder things to really wrap your head around, because it sounds like three things, but they sound almost the same. Scan to scan, intrasample, and sample to sample. Like, what does that mean? So scan to scan is literally from one scan to the next scan, how much do those ions, how much do the masses shift for a given ion? From intrasample, it's not of the same ion, but it kind of goes back to that we have C12 and then we have C13. How much do the masses differ within a single sample of the C12 and the C13 within the same sample, within the same molecule? And then you have lastly sample to sample. So now we're talking about different injections. How much do the masses differ from one injection to a next injection of comparing the same molecule? So that is good. Next, we have some kind of filters. You're going to get thousands of metabolites. Maybe you want to filter some out. You can remove each feature list. Please change it to keep, and I'll show you why, and why I think it's good to keep it, especially when you're first doing initial analysis. Minimum aligned samples. So we are running everything in triplicates of the same species. So we expect if we find a metabolite in one sample, since we're doing biological replicates, 3 different leaves, we expect the same metabolite to be found in the leaf each time if it's coming from the same species. So what we could put here is 3. So let's say for some reason you have in one of your 3 leaves replicates, you see a peak, but then it's not found in the other 2, it will get removed. Because again, we're using triplicates, we can put that we want to see it 3 times. And again, maybe you're only using 1 replicate, which is fine. Maybe you're doing a large screening of thousands of samples. You can keep it at 1, but today we'll use 3. This is, again, the same concept, but looking at the metadata column. You do not need to worry about that right now. This is if you're using pooled QC, another filter. We're not using that right now, so we'll not go into that. This is the filter. Let's say you have a peak, but that peak doesn't have a corresponding C13, so isotopic peak, maybe it's a lower intensity metabolite, it will get discarded. We don't want to do that, so we'll keep that unchecked. And the last thing, apply strict shape filtering. So ideally, our peaks should be relatively Gaussian shaped. We could put a filter that says, hey, is this peak a nice shape? Yes, keep it. No, get rid of it. Today, we already know all our peaks are going to look amazing, of course, so we will not check it. Excellent. Next is annotation. This is probably, as Tomasz mentioned, one of the stronger points of Mzmine. I just want to kind of go back to what I said in the beginning. We are using data-dependent MSMS. So every once in a while, we take a metabolite, we fragment it into smaller ions, and those ions have key structural information about the structure. We can actually take that fragmentation and compare it to fragmentations of purified standards or libraries of reference spectra and compare them. So what does that mean? What does that sound like? So just to kind of visualize it a little bit better, let's say you have one molecule and it is molecule A. It fragments one way. Let's say you have the fragmentation of the same compound. You know it's this compound because you purchased it as a standard. You fragmented it. You have another spectrum. You can convert it into a vector. It's very standardized math. You don't need to understand it completely, but just know this has kind of been used for a long, long time. And you can then compare the vectors of the 2 fragmentation patterns. If the vectors overlap, great, that means your spectras overlap. You have most likely that molecule found in the library. If the vectors don't overlap, then it's likely that the spectras aren't very similar, and you do not have the molecule that you are trying to find. And Tito will kind of go over this a bit more, what really is the MSMS, the fragmentation, and how this kind of works with the molecular networking in the next session. So, with that being said, we need to import a library file. So again, please go to Select Files. And one of the files I provided you was this Mass Bank of North America MSMS library. So basically what this is, again, is just a library of fragmentation spectra that correspond to known molecules. So you have one molecule, one fragmentation, it's one entry. You have another molecule, another fragmentation, another entry. And we can use this file to explore the fragmentation of the unknown molecules in our sample. Local compound database search. So let's say you have some masses you're interested in, and with known retention times, this can be used to then just search for those molecules that you might have a list of. Annotate lipids, we will keep checked for now. That really depends on if you are interested in lipids or not. And we have a formula prediction, but we will not go over that today. While I showed select files, you could always just drag and drop. A lot of the times I'm showing you with the button, but often, anytime you could import something in Mzmine, you can just drag and drop. And lastly, some of the I think, most important things you could also do in Mzmine, molecular networking. We want that checked. And then we want an export path. So what we want to do is, what Mzmine does really nicely is once you process our data, it exports it all to the same folder with all the outputs you need in one convenient place. So what I will do is I'll select the folder, I'll go to desktop again, and I will make a new folder for the thing called Okay, I'll just use this output folder and output folder, and I will name this KMOS, Korean Metabolomic Society, Mzmine, workshop. I'll save, and it will update here in a second. And I will also have export for molecular networking checked, export for serious checked, and I will not select export annotation graphics, it will just slow it down. So once everything is filled out, I will press create batch, and boom, it makes this nice batch that I can process all my data, and it already has everything filled out. So it will import the data for you based on what you selected, it will import the metadata for you based on what you selected. It will import the spectral library for you based on what you selected. It will do all the steps, and again, there's a lot of parameters that we didn't cover, but the wizard extrapolates the best parameters based on what you put there. And again, the best thing you could do at the end of the day is just play around with these once you kind of get more familiar with them and see how it changes your data from one thing to, from one setting to another. So, what I'm going to do now, we're all going to press OK, and you will see in the bottom, it is now doing its thing. One thing I forgot to mention, I am very sorry. If you didn't select a location for in set preferences, there is a temporary file directory. If you didn't set that, it might be a bit slower in the data processing. I'm sorry, I forgot to mention that in the beginning. Usually when you set this, do it somewhere local on your computer. If you set this somewhere like on a cloud, it can really just become buggy, maybe crash the software because it's trying to upload temporary files to the cloud and it's taking way too long. I have mine set to my desktop, so if it didn't have it set, hopefully the default location that it has is fine, or sometimes it will just put the temporary files as like a quick temp folder of its own. And depending on how fast your computer is, you might be finished. It says 2 tasks left, but sometimes there's no content left. I can tell you that's just thinking it still has 2 tasks, but it's done. If you weren't sure, if you open up the tasks tab, you will have something listed here still. So, in the end, you now have your feature lists. And why I said keep, it gives you a better idea of what we just did. So for each sample, what we did was we built an XIC for every metabolite that we could find. So again, an XIC is just the tracing of a given MZ over a retention time. And we can't create an XIC manually, hand by hand. It would be just very exhausting. So what we did was we first built an XIC for each sample. We smoothed the XIC. We split the XIC. So let's say, for example, these are 2 isomers, but we don't want them as actually the same feature entries since they have 2 different retention times. So in the next steps, we did local minimum resolver. So we took those signals that were once in the same chromatogram and made them their own entries because they have their own retention times. And basically, what we did was create our feature list. Each one of these is a metabolite in the sample, a feature, metabolite, small molecule. Again, these are all synonymous. And what we did afterwards is we had it built for every sample, but we don't, again, now we have about, let's see if we scroll to the bottom. I don't think we already said feature ID. We have probably close to 1,000 small molecules detected in this sample, but we have 15 samples. So again, it's not really realistic for us to look at 15 files with 1,000 metabolites in each file. So what we did was we joined the files together to create our final feature table. This is not the final one because we did some other filtering, but what this is now, each row represents 1 metabolite in the sample, and that has a retention time. So how long did it take for the metabolite to reach the detector, the feature to reach the detector, the M over Z, and then it has the intensity for that sample, for that intensity found in each sample. So you can see right here, this metabolite at 1.24 minutes is found in every sample around the same intensity, 2E7, 1E8. If I see something like that, I'd probably think either one of two things. Maybe it's a primary metabolite found in all pipper species, or maybe it's some background contamination that was just found because it was just accumulated during the sample processing. And ultimately, we created this final feature table after filtering steps and spectral library matching. And you can see we have some annotations. So this just kind of goes back to highlighting that we can take an unknown metabolite. Before, this didn't have any annotation. And then we took the fragmentation of that molecule and we matched it to the library fragments as well. And just so you can see what that looks like, you can right-click anything that has an annotation. You can go to Show, and you can select Spectral Database Search Results. So just so you can actually see what is going on, I can't see from this angle. Okay. So the bottom, this fragmentation spectra is the spectra from the library. So this fragmentation is due to some standard. Mass Bank of North America took the standard, fragmented it, and annotated these fragments corresponding to this molecule. We had an unknown molecule in our sample that had this fragmentation pattern, and it aligned with a cosine similarity score of 0.935. That's pretty good. And since we know this is a pipper plant, it's very likely that it should have this pipper-related alkaloid. And we can use our expert knowledge. It's a pipper plant. This pipper alkaloid has been reported in pipper species before. There's a good chance that this is actually the molecule. But at the end of the day, the best thing to do is confirm it either with isolation and NMR or with a purchase standard and just run it yourself on the instrument. With that being said, this, one last thing I want to show. Before you set in the Mzmine wizard the path, and you now have all your results exported into a single place. And this is really good for later analysis. So let's say you analyzed your samples 3 years ago and you forgot where you named it, you forgot where you put one file. This puts all the files in the same place. So it's really hard to lose. I really wish this was here when I started my PhD because some of those files were spread out. Now there's no reason why it shouldn't be. And this makes it really nice for publication time because we really want to follow fair practices. So really making the data findable, reusable, and accessible for other users. So I can now easily upload this as attachments in a publication or as a Zenodo file. And what this is the different exports you can use for different post-processing tools. So right here is a GNPS MGF, so unfortunately we won't go into that today, but you could use this file to analyze your feature table on GNPS or GNPS2. You also have another MGF file for Sirius, so if you want to predict the structure with Sirius, or if you want to predict the compound class with Sirius, you can use this file. You also already have a molecular network file from Mzmine, which is found right here. Teacher will go a bit more into the molecular networking here in a minute. Anything else? You also have the metadata. Oh, the most important thing of it all. It exports the batch file. So let's say you don't remember what settings you used. You're like, I need to reprocess the data. I reanalyzed a couple samples that for some reason didn't look good, but I don't remember what settings I used. This batch file are all the settings that were used to get to these results. So let's say I go back to batch mode, and let's say I accidentally removed 2 steps, and you don't remember what 2 steps you removed, you don't remember exactly where they were. I can just go to load. I can go to desktop.